Welcome to the next lecture in Bio 120. Today we're going to be having a look at how hosts and pathogens interact, in particularly in relationship to vectors. So we're going to be looking at diseases that are transmitted by vectors, and vectors are defined as a carrier, usually an arthropod, either a fly, a flea, a tick, or a mosquito, that is able to transfer an infectious agent from one host into another. So we're going to be looking at two vector-mediated diseases. We're going to be looking first at malaria and then at the bubonic plague. So malaria, here is the introduction to it. I will leave you here with the fact that it's transmitted by the mosquitoes Anopheles, who you can see here taking a blood meal, and later you can see why I'm talking about Jack, which I have over here for you. So malaria, it's caused by an obligate intracellular parasite of the genus Plasmodium. As you remember from what we had discussed earlier in eukaryotic movement, Plasmodium is also an Apicomplexa. And the vector for this disease is going to be the mosquitoes of the Anopheles family. There are about 400 species, of which 30 are vectors for malaria. Now, we are the only disease reservoir, and a reservoir is defined as a habitat in which an infectious agent normally lives and grows and multiplies. So we're going to see that there's habitats that are reservoirs that are human, either animals or some of them are actually environmental. We're going to look at 4 different species of Plasmodiums: Plasmodium vivax, falciparum, malaria, and ovale. We're going to concentrate mostly on falciparum because it is the most dangerous of them. And the last that I want to bring you is a new Plasmodium, Plasmodium knowlesi, which usually causes malaria in macaques, but has been recently shown to start to infect humans. Now, when we think about malaria, there's about 4.3 billion people that are living in areas that are at risk for malaria in about 106 countries. The countries in green are countries where malaria transmission is not known to occur. The countries in yellow are areas which have malaria transmission, and the countries in red are areas that malaria can occur. As you notice, there is a band in the middle of the planet where malaria happens, and it's usually the tropical and subtropical areas. Now, the World Health Organization is estimating that malaria caused 207 million clinical episodes of malaria, and that of those, 627,000 deaths happened. Of those, 91% of the malaria deaths happen in the continent of Africa. Now, malaria is the 5th cause of death of infectious diseases in the world. It's second after HIV in Africa. Now, what I'm going to show you over here are red blood cells that are infected with Plasmodium vivax. These are human red blood cells. And now this is where I bring Jack. Any similarities are purely coincidental. We're going to be looking at now some of the biology of these Plasmodiums and see how they differ according to their behavior as well as geographical location. So what we can see is that in this table that I got from review of clinical microbiology from 2002 from Carter and Mendes, it's showing that of the 4 different kinds of malaria, Plasmodium vivax is the most common. That is the area over here shown in red. It causes about 95% of all the cases in Asia, 87% of the cases in Central America and the Caribbean, and it causes about 70% of the cases in South America. Plasmodium vivax, it's not a very important pathogen when we come to talk about Sub-Saharan Africa. That is actually Plasmodium falciparum. And Plasmodium falciparum causes between 88% to 78% of the cases in Sub-Saharan Africa, with about 30% of the cases in South America. Very little of them in Asia, and about 12% of them in Central America and the Caribbean. Now, Plasmodium vivax, it's the most commonly distributed of those parasites, and it causes the relapsing malaria. Plasmodium falciparum, which is found in tropical regions, causes the most severe and fatal kind of disease, which is the one found in Sub-Saharan Africa. Now, Plasmodium malariae, it's the 3rd kind of Plasmodium, and it doesn't cause disease in Asia, Central America, and very little in South America, mostly the disease is being caused in sub-Saharan Africa, and it is also a microorganism that causes the relapsing malaria. Last is Plasmodium ovale, and Plasmodium ovale, it's the least abundant of it. It doesn't cause disease in Central America and the Caribbean or South America, very little in Asia, and about 8% of the disease of Plasmodium ovale is caused in Sub-Saharan Africa. So it's the less common malaria species, and it causes, again, relapsing malaria. So this shows the 4 geographical locations that, this shows the 4 different kinds of Plasmodium and the geographical distribution that they have. So Plasmodium falciparum and Plasmodium vivax are the 2 most important pathogens in this group. Now, the disease is different according to the organism that causes. So I'll recommend that you visit the Center for Disease Control or the World Health Organization websites on malaria so you can get more direct symptoms information about these pathogens. But what I want to show you is that there are a set of steps that the pathogens take, and they can cause what is called an uncomplicated malaria. That is what we consider to be classical malaria, but is rarely observed actually in the clinics. And this comes as an attack that lasts between 6 to 10 hours. It has 3 different stages. A cold stage, which the person has a sensation of coldness and shivers a lot. Then you have a hot stage, where you have now fever, headache, vomiting, and seizures in small children, as well as a sweating stage, where the person then sweats and they return to normal temperature, but they remain very tired. Now, these are usually occur depending on the Plasmodium, either on the second day with the tertian parasites, which are Plasmodium falciparum, vivax, and ovale, or on the third day, which is the Plasmodium of the quartan parasite, which is Plasmodium malaria. Now, in reality, what patients present is a different combination of disease, including fever, chill, sweats, headaches, nausea and vomiting, body aches, and the general feelings of illness. Now, malaria becomes really complicated once it goes to the stage of severe malaria, in particular when malaria goes to the brain, which causes abnormal behavior, impairment of consciousness, and induces seizures. People often fall in coma and they develop neurological abnormalities. Because the parasites are infecting red blood cells, they kill them, and therefore they can also induce anemia. And out of the anemia, now you have hemoglobin that is found in the urine, and that condition is called hemoglobinuria. Now, depending on the parasite, they can also cause an acute respiratory distress syndrome, which is an inflammatory reaction of the lungs where you have the Plasmodium infecting the red blood cells that are present in the lungs, and therefore they reduce the oxygen exchange that the lungs have. Because they are destroying red blood cells, you have abnormalities in blood coagulation as well. Blood pressure comes down, and because of the many different parasites and the debris that the red blood cell destruction happens, you can have acute kidney failure. Now, they cause a state of also hyperparasitenemia, which is about 5% of the red blood cells now infected with malaria parasites. What we're going to see is that depending on the parasites, you can have metabolic acidosis, which is acidity in the blood and tissue, which is associated also with a hypoglycemia, which is low blood sugar. Now, severe malaria, which is the one that is actually caused primarily by Plasmodium falciparum, it's actually a medical emergency and should be treated immediately because it can cause death. So let's take a moment to look at how the Plasmodiums are infecting their host and some of the different stages that infection occurs. So we're going to start by introducing you to some of the stages of parasite infection. Now, we're going to be looking at Plasmodium falciparum for the majority of this slide. And what I want to bring you to the idea is that this kind of parasites has very different forms according to the stage of the infection. And the first form that we're going to talk about is going to be the sporozoite. The sporozoite is the infective form that is passed from the mosquito Anopheles to the human host. Now, shown over here to the right are these very long cells with one nucleus, which are the infective form of the sporozoites. Now, the sporozoites are able to invade the liver of the person after they have been transmitted from the salivary gland of the mosquito through the blood meal into the circulation. Once they are in the liver, after days of infection, they can undergo a metamorphosis into a stage called the merozoite. So the merozoite, it's a stage in which now allows for the movement of the parasites from the liver into the blood. And the merozoites are the parasites that are able to infect the red blood cells. So this picture here on the right, it's showing some normal red blood cells as well as some red blood cells which have burst after they have been infected by the Plasmodium. These are called a schism, and the schism is able to burst open like this one over here, releasing the merozoites that are able then to infect other red blood cells. Now in the red blood cells, the parasites are going to undergo asexual reproduction that is going to allow them to divide and therefore release more parasites once they kill the red blood cell. And here is when you have the resulting anemia coming from red blood cell damage. Now, at a certain point, some of the parasites that are infected red blood cells are going to differentiate into gametocytes, and these are the sexual cycle of the infection. You're going to have both male and female gametocytes that are going to allow for the reproduction, and they're going to be then released into the environment. So here in this image, you have a male parasite to the left and a female parasite to the right. However, the mosquito is going to take a blood meal and take those gametocytes. And in the mosquito, now those gametocytes are going to fuse into a zygote. So the zygote is going to be now the reproductive form of the parasite in the mosquito, and it's going to go from the gut, and in the gut form a different distribution state, which I'm going to show you in a moment. So now we're going to be looking that you have two differentiated stages. One differentiation happened in the human host, and a second differentiation happened in the mosquito. Now this image here from a paper from 2004, it's showing how the sporozoites are being injected through the salivary canal of a living salivating mosquito. So this is a movie. My colleagues at the Pasteur Institute used to work this, so I got this from one of their papers. So what you can see are these lines which are moving from this area of the proboscis to this area here at the end as the mosquito salivates. So as I put them out, you can see the lines of the sporozoites which are passing through the saliva into the proboscis, and later they're going to be injected into the unsuspected host. So what I'm going to show you here is the life cycle of the parasites, starting from an infected Anopheles mosquito that is taking a blood meal from an uninfected patient. So now, this mosquito over here, it's taking a blood meal. It has sporozoite in the salivary gland, and as I show you in the previous movie, it's going to be able to inject those sporozoites into the host during the time that it takes a blood meal. Here are the sporozoites, these elongated forms of the parasite, and they're going to move through circulation into the liver, where they're going to infect liver cells. In the liver, they're going to be able to form a schism, and the schism, it is a liver cell that is infected with the sporozoites that are now differentiating. Depending on the species, for example, Plasmodium ovale and Plasmodium vivax, they can have another state in which the sporozoites remain inside the liver cell, forming a cell that is called a hypnozoite. The hypnozoites are long-lived and can stay for many years into this undifferentiated state, allowing for the clinical relapses that can allow the mosquito to, the parasite, to develop again. So I think I said years. No, actually, I meant weeks to years. Yeah. So it can be dormant for weeks, from weeks to years, causing this clinical relapse. Now, eventually, the schism, it's going to burst, and when it bursts, it's now going to release a different type of Plasmodium into the bloodstream, and that's going to be the merozoites. The merozoites now that are flowing in the blood are the part that is going to cause the erythrocytic phase of the disease. The first phase of the disease is the exoerythrocytic phase, which is happening at the liver, and the second phase of the disease is the erythrocytic phase, which is happening in the red blood cells in blood. So what we have over here are Sporozoites, which are now infecting a healthy red blood cell. They're going to begin to divide asexually until they form another schism, and in the schism is going to eventually be able to burst to release more Sporozoites, continuing with the cycle of infection. And this is the part that causes severe anemia. Now, as we are with a certain level of frequency, an infected red blood cell is going to allow the cells to differentiate into gametocytes, and those are going to be both the male and female gametes, which are able to be taken by a brand new mosquito when that mosquito takes a meal from an infected malaria person. Now, in the gut of the mosquito, the gametocytes are going to form a zygote. The zygote is going to differentiate into an okinet, and the okinet is the cell that is able to infect the mosquito having sexual reproduction in the mosquito midgut. Eventually, you're going to have movement of those parasites from the midgut of the mosquito into the salivary gland, where now they can infect salivary gland epithelium, creating more sporozoites. And the cycle repeats again when this now newly infected female is able now to bite another unsuspected malaria victim. So now in this slide, I have the text that accompanies the previous figure. So I'm going to leave it over here for you to take. The numbers around are describing the images in the previous image, the steps of the previous image, so you can now have a better idea of what's going on and be able to then review the life cycle of the Plasmodium. So in reality, most of the Plasmodia have a very similar life cycle, but in this case, I concentrated on Plasmodium falciparum. So now let's take a look at how malaria evades the immune system. And malaria, it's a master cell of evading the immune system because it has a protein on its surface called the Plasmodium falciparum erythrocyte membrane protein 1, or PFEMP1, which is a molecule used on the surface of the red blood cells to attach to the Plasmodium. Now, there are about 60 variable genes of this protein in the haploid genome of a Plasmodium. And the persistence of the Plasmodium falciparum in the blood stage, is going to depend on the successive expression of this protein in different variants that are going to be designed to avoid the immune system through the process that we discussed already to be called antigenic variation. So each individual parasite is only going to express one variable gene of the PFEMP1 at a time, and is going to maintain the other members of the family of proteins in a transcriptionally silent state, and that is done through an epigenetic mechanism. Now, once you have the variability of this protein, is going to enable the parasitized red blood cells to adhere to the macrovasculature, resulting in severe disease. There in the macrovasculature is where they're going to stick, they're going to develop into a schism, and they're burst. Now, different ligands mediate adherence to different kinds of receptors in endothelial cells. So, for example, some of the PFEMP1 can bind to the scavenger receptor CD36, other ones bind to the chrondoin sulfate A, and other ones bind to the ICAM1 protein. So together, the antigenic variation that is happening to the different stage of the blood-borne disease of malaria allows the parasites to escape immunological pressures mediated by antibodies. The other thing that is relevant for this parasite is the fact that they are becoming resistant to the anti-malarial drugs that we're currently using, which are quinones. Now, malaria happens in regions with really poor conditions that promote disease. Here are pictures from a clinic in East Timor, and as you can appreciate, the patients are living in poor conditions. They have mosquito nets to prevent infection and exposure to mosquitoes, and once a patient is infected, they only are able to treat the conditions. As more eventual malaria drug-resistant mosquitoes begin to occur, the less likely that the patients are able then to survive the disease and then die. So one of the things that I want you to keep in mind when you look at the lecture and we come in discussion tomorrow, it's the triangle of disease and how the different components of the triangle, the host, the environment, and the parasites, could be modified to prevent disease. Now, let's talk about the other disease that I would like to introduce you to, and that is going to be the bubonic plague. And the bubonic plague, in this image, I am showing you 3 different images. They are all from artwork. One of them over here is showing this painting describing a European city that has been ravaged by the plague. Here, this medieval painting has a doctor who is performing some incantations to cure the patients from malaria, from the plague, excuse me. And what you can appreciate from the people in bed, both men and women, is that they have this really large areas of inflammations all over their bodies, and those are called the buboes. Here is an Italian, this is the costume that was worn by Italian doctors during the time of the plagues, and they were having this mask, and the mask allowed them to put herbs and other aromatic compounds to basically deal with the stench of death that was pervasive during the time that the plague attacked. So now, the Black Death, as it was called, is considered to have caused about 50 million deaths across Africa, Asia, and Europe in the 14th century. And we believe that it wiped out about half the population of Europe in a really short amount of time. Now, there was an outbreak in London that we call the Great Plague of 1665, and that killed about a fifth of the population of the city of London. And then in the 19th century, the Great Pandemic of China and India is believed to have killed over 12 million people. Currently, the bacteria that causes the plague are endemic in different regions of the world, as shown here in this map, including Madagascar, the Democratic Republic of Congo in Africa, Peru, China, and the Western United States. So now, the plague is caused by an infection with the gram-negative bacteria Yersinia pestis. It is transmitted through fleas, and the fleas are of the Xenopsila chiopis species, and it's called the oriental rat flea, which is usually found on rats of the Rattus rattus species. Now, the flea is the primary vector that transmits the plagues in most of the plagues that happen in Asia, Africa, and South America. And both genders of the flea are able to transmit disease because both of them take blood meals. Here in this image, you can see the Yersinia pestis parasites, and as you can see, it is a small rod that happened to occur in a dibacillus kind of morphology, so usually in pairs. Now, we believe that there are 3 forms of the plagues that have different kind of symptoms of the infected person. And the first one is going to be the bubonic plague. And it is called bubonic plague because it refers to the appearance of these very painful lymph node swellings called the buboes. This is the classical sign of the bubonic plagues, and the buboes appear in the area of the groin, the armpits, and the neck. And once infected, those buboes are going to ooze pus and blood. The mortality rate of the bubonic plague is between 30% to 75%. And the symptoms are fever, a very high fever between 38 and 41 degrees. Patients undergo also headaches, joint pain, nausea, vomiting, and a general sense of malaise. Now, patients that have the bubonic plague can pass it to other people once that plague goes systemic. And here is the septicemic plague. Now what we have is bacteria present in the blood. And these are cases that are going to lead to about 100% mortality. Now, once the bacteria is in the blood, it's going to cause really high fever. The skin is going to start to turn purple and black due to what is called disseminated intravascular coagulation, of DIC. Now, as it goes into the blood, it can then pass into the lung. And here is the stage in which you can have an airborne plague that is passed from person to person through saliva and coughing. And the mortality rate of the pneumonic plague is between 90 to 95%. Now the person, besides the symptoms that I described before, are also going to have slimy sputum that is tinted with blood. And as the disease progresses, the sputum amount becomes more copious and bright red because of the destruction of tissue in the lung. Now, the victims that are having the plague are going to have the dark blotches in their skin, and that is called acral necrosis. And it leads to the name of the Black Plague, as those black areas become to cover the skin, oftentimes turning black, and that is what gave the name of the plague, the Black Death. Now here in this image, we're showing a couple of the symptoms. So this person here has a very large enlarged buboes on the area of the groin, and this young woman has a very large swelling on the neck. So there's a buboes on the neck and the groin. This is also showing here this hand has subdermal blackness due to the hemorrhage that we talk about. So these are the three major visible symptoms that could be attributed to the plague. So now the plague can come and have a set of 5 different stages in this life cycle. You have the first, which is the urban cycle, and this is an explosive pandemic disease with the high mortality, and in the 6th century it killed about 100 million people and about 25 million people in the 14th century. And it's the urban cycle because it's happening in urban conditions where rats are able to pass their flea to people, and then the explosion goes from person to person. But when it goes to person, it's when it becomes the pneumonic plague, as I mentioned to you earlier. Now, those are when the person begins to cough that have droplets that contain bacteria that fly across and infect other people. Or it could also be caused by fleas biting other individuals. We have not had a case of the pneumonic plague in the United States since 1925. Most of the plague is maintained in a sylvatic cycle on reservoirs, and that plague, it's the type that we have been seeing. So it's basically contact between people and animals in the wild through their ticks that are able to allow transmission of the bacteria. So for example, there was a wildlife biologist called Eric York who died in 2007, of pneumonic plague, and that person was working in the National Park Service in Grand Canyon in the area of Arizona, where he got exposed to the plague. So this is the first case of pneumonic plague that we had seen since 1925. Now, you also have the fact of enzootic plague, and that is the plague in which you have a resistant host, a host that can be infected by the bacteria but will not have disease or have very low profile and stable disease, and therefore you have a reservoir, and those are voles and deer mice, and those are part of the sylvatic cycle. The other part of the sylvatic cycle is the epizootic plague, and those now are going to be sensitive hosts, basically animals like prairie dogs, road squirrels, and rats that can fall prey to the disease and are able to pass it to other animals. And that results in a high visibility of disease because a lot of animals begin to die because of the epizootic plague. Now, let's take a look at how all these 5 different cycles are related to one another. So what we have over here in this image, it's here the sylvatic cycle in which you have some kind of wild rodent that is infected and is bitten by a flea, and the flea is able to pass that disease to another animal that is also now going to become infected. You can also have exchange between animals, as shown here by this animal that's other. You can also have exchanges, and we hypothesize that it's either rare or theoretical, that wild animals can pass it to other animals by contact. But the direct contact route is going to be the exposed animal generating infective fleas that are going to pass it to another host. And therefore the sylvatic cycle is maintained. Now the fleas are able to interact, for example in here, with animals that are present in the urban setting, like the rat. So you can have fleas that come in contact with rats. The rats are now going to become infected, they're going to have fleas, and they're going to be passed along other animals. This generates the urban cycle. Now that you have a reservoir of animals infected in the urban cycle and a reservoir of animals infected in the sylvatic cycle, you can have a direct contact to people, because people are either hiking in the sylvatic cycle areas in the forest, or they are present in cities where the animals, like the rats, are infected. So you can have now direct biting of fleas, either because people are directly contacting the rodents or the fleas are biting them. In either case, now you have individuals that develop the bubonic plague. Now, if not treated, the bubonic plague is going to progress to the secondary pneumonia, which is the pneumonic phase, shown over here by this person with infection in the lung. And now you can have direct person-to-person contact to generate the pneumonic plague epidemic that it's very lethal. Now, when we think about the United States, there have been many cases of, very incidents of the plague in the United States. Since the year 2000, there have been many different cases, alone 15 of them in 2015. Of those 15 cases in 2015, 4 people have died. So the closest one that I can remember was this gentleman over here called Paul Gaylor, which is from Oregon, who was apparently bit by his cat. And his cat was in contact with some of the animals in the rural area where he lives, and he developed the plague, as you can see over here. You can see the symptoms of his hands turning completely black. The gentleman was on a coma for about a month and eventually managed to pull through. Today he's still alive, but they had to amputate part of his hands to eliminate the area of necrosis caused by the extensive hemolysis in his blood. What you can see from the map over here is that the majority of the cases for the plague are found in the western region of the United States. So here are the 4-state area of New Mexico, Arizona, Colorado, Nevada, California, Oregon, Idaho, and Montana. So most of these areas over here, each dot represents a case of exposure to the plague. This one here in Illinois happened from people who came hiking around the Grand Canyon area. So that is not, that is caused by travel. So this is one of the things in which travel can actually be able to help the spread of a particular epidemic. So when we think about this particular bacteria, it has many tricks under its proverbial sleeve to avoid the immune system and induce infection. So one of them is the capacity for adhesion and invasion, and it contains Plasmodium activator protein that is going to facilitate the invasion of the bacterium with the extracellular matrix of the host tissue. It also has ways to acquire iron, and that is going to be by the presence of the Yersinia bactin molecule that is an iron siderophore, so it's able to acquire iron from the host directly. Now, importantly, it has type 3 secretion systems, and those type 3 secretion systems, as we remember from the lecture in carbon, are designed to secrete proteins from the cytoplasm of the bacteria into the cytoplasm of a target cell. Through the type 3 secretion systems, this bacteria is able to directly transport Yersinia outer proteins, or YUPs, that are able to have outcomes of disease. And this type 2 secretion system, as well as the YUP, are encoded in a Yersinia virulent plasmid called BPYV that is encoded only on the strains that cause disease. Now, the YUP effectors are a family of proteins that have many diverse functions. They can disrupt the acting macrofilaments to avoid, for example, the phagocytosis of macrophages. Some of them are able to migrate into the nucleus and interfere with the cell cycle. Other ones are able to inhibit the MAP kinase system and the NF-kappa B signaling pathways to prevent activation of immune cells. Other ones are going to directly cause apoptosis of macrophages and neutrophils and able to kill them, so they do not allow the immune system to target the bacteria and therefore have problems inducing active inflammation. And last, because they are also expert at avoiding the immune system, they have anti-phagocytic antigen factor 1 and V antigen, which are going to prevent the phagocytosis of Yersinia pestis by macrophages. So now all those virulence factors are giving the advantage to the bacteria so it avoids the immune system. Of this, the most important clinically we consider to be the type 3 secretion system. And as I mentioned to you, this is a complex of about 42 genes that encode an entire syringe-like structure that is able to pass proteins from the cytoplasm of the bacteria into the cytoplasm of a host cell. Now, as I mentioned, this is encoded on a plasmid, and the plasmid also encodes the yolk proteins. Now, as I mentioned, the yolk proteins can help in prevent phagocytosis. They are able to attenuate the phagocytic oxidative bursts that are going to generate the antimicrobial reactive oxygen species. They're able to downplay the inflammatory response caused by the macrophages, and they're able to kill natural killer cells, dendritic cells, and neutrophils. So just to remind you, here to the right we have a picture of a type 3 secretion system, which is called the injectisome. It has a lot of similarities to the proteins that are encoding the flagellum, shown over here in the left. And because, like the flagellum, it is a hollow structure that hydrolyzes ATP and is able then to transport proteins from the cytoplasm of the bacteria into the cytoplasm of a target cell. So here at the bottom we have the membrane and the cell wall of our gram-negative Yersinia pestis cell. And to the right, we have now that when the type 3 secretion system encounters another cell, the stop valve that is preventing the movement of cell components from the bacteria to the outside is blocked, and only when you have a conjugation between the between the Yersinia pestis microorganism and a target, are you able now to use ATP to pump across those effector yolk proteins into the cytoplasm of the target cell. From this review in Nature Medicine from 2005, I am here showing you again the Yersinia pestis microorganism shown here in red, and here in yellow we have a putative yolk protein. That yolk protein will be transferred by the type 2 secretion system into neutrophils, macrophages, and dendritic cells and natural killer cells, and they are going to allow them to be killed by apoptosis. You have basically wiped out the cells of the innate immune system that are able to be the first defense against bacterial infection, and you only have left behind B and T cells, which are part of the adaptive immune system. And without the dendritic cells and the macrophages, the adaptive immune system it's not able to effectively launch an immune response against the bacteria. Therefore, by crippling the cells of the innate immune system, you have inactivated the adaptive immune system. That is going to now allow the bacteria to be able to proliferate and cause disease that eventually lead to the person and the host death. So that is the last topic that I'm going to talk about in this lecture. What I have here at the end are other slides. This one in particular is a more complicated slide from the Center of Disease Control describing the malaria life cycle. And the next slide following to it are, again, each of the steps present. It's described here in a little bit more detail, talking about how it is affecting host infection. So I will leave you with this, and I will then see you on Tuesday. Bye-bye.